very, very pleased to welcome Phillip Domeier, who did his grad work at Penn State University in a globally famous town that is home to Chocolate World and the giant roller coasters at Hershey Park. And he's a Pennsylvania boy. He did his his PhD characterizing B cell receptor signaling and regulation of autoimmune germinal center. And then he kind of continued that knowledge base as a postdoc in Seattle at Benaroya Research Institute, where he ventured further into B cell and germinal center regulation. And really, that's what you're going to continue telling us about, where only about a month ago he joined UBC as an assistant professor. And I'm really excited to have met him at a distance yesterday, where I saw him through a window where I was sitting warmly in blends at about 8 a.m. and he was snuggled in about 4 layers of clothing as he scurried by to catch the bus. So welcome, Phillip.
Thank you for the kind introduction. I guess you guys can hear me because I can hear me. Echoing? Okay, we're good. Well, thank you for the opportunity to kind of talk about what I am excited to start at UBC, but also to talk a little bit about one of my favorite areas of study, which are these structures called germinal centers. And I kind of showed the same picture kind of inverted twice to kind of give you this idea that these structures are actually can be very helpful in generating antibody-mediated responses after infections or vaccination. But if they're not properly regulated, they can also become quite a problem where they can generate self-reactive autoantibodies and drive autoimmune disease. And so we look at both sides of these structures to kind of find these new novel pathways we can use for different therapeutic interventions. And so I'll talk about in this particular talk 2 projects I did as part of the Benaroya Research Institute. Don't lean into it. That tie into both of these different features. And so the first part, I'll be talking about how these protective germinal center responses can be augmented in the context of vaccine development by targeting this particular cytokine called thymic stromal lymphopoietin. And then in part 2 of this talk, I'll be talking about some autoimmune germinal center responses, notably how they can mediate the production of autoantibodies after UV photosensitivity reactions. So first going into this, what the germinal center actually, how it works and what it does. And so upon B cells and T cells interacting in secondary lymphoid organs like the spleen and lymph nodes, they find their cognate helper cells, and these B cells that activate their cognate T cell have a weak affinity for antigen initially. So these naive B cells are broadly reactive. And so they can undergo this T cell independent plasmablast pathway where they can secrete IgM, or they can undergo this more dramatic transformation in the structure called the germinal center. So in the germinal center, those B cells that have this kind of moderate reactivity to antigen actually rapidly clonally proliferate. And what's exciting is each one of those cells undergoes this process called somatic hypermutation, in which they can modify the portion of the B cell antibody gene that binds the antigen of interest. And so with these modifications, you derive a very wide variety of antigen affinities out of these B cell populations. So you can get higher affinity binds than the parent clone. You can get moderate binding affinity interactions, or even low or non-reactive mutations. You can also get self-reactive mutations in this process. Again, these are random, or mostly random, so they can actually generate novel autoreactivity in the germinal center. And so we need to select out those good B cells, the ones that provide us the highest affinity interactions from the cells that are going to cause disease. And so this process is mediated by several different cells. The 2 notable players that I'm going to talk about today are these cells called follicular dendritic cells, and they capture immune complexes on their surface that contain the antigen, kind of like holding up targets for these B cells to collect. But the interesting thing about these structures is that they had a limiting quantity of antigen on the surface. So the highest affinity binding B cells compete for a limited antigen availability. In addition, those B cells, after acquiring the antigen, must then present the antigen to a helper T cell called a T follicular helper cell to acquire survival signals and activation signals so they can become cell that escapes the germinal center. So the highest affinity, the B cells that get the most T cell help, most likely become plasma cells, but they secrete antibodies at a high concentration and move out into the periphery. Moderate affinity mutations often generate into these germinal center derived memory B cells. And we now have marker profiles we can use to identify memory B cells that have transited through the germinal center process versus ones that have been activated independent of the germinal center process. Lower affinity B cells can actually recirculate to the germinal center. They can do another re-roll, try again, make another mutation, potentially get a higher affinity mutation in the process. But self-reactive or non-reactive B cells should undergo apoptosis and be cleared from the system. And so this is modulated by macrophages in the germinal center. But if you have defective clearance of these apoptotic cells, this can drive autoreactivity. Another player we're going to talk about a lot today are these regulatory T cells called T follicular regulatory cells. And they are kind of newly identified in the past few years. And they play this complex role in both blocking the germinal center expansion through inhibiting B cell proliferation and mutations, but they also can play a role in controlling the stringency of selection. And so having these Tregs inside the germinal center actually helps to get higher affinity B cells because it sets the brakes, the upper limits, and prevents these germinal centers from going out of control with these mutations. So they're all playing a very careful and well-balanced role in this germinal center. And there are multiple ways you can actually disrupt the germinal center to elicit higher affinity mutations that help us, but also can drive autoimmunity. So we're going to first talk about a cytokine called TSLP, thymic stromal lymphopoietin, that we know is released by these follicular dendritic cells in the germinal center and play a role in the selection process. So thymic stromal lymphopoietin, TSLP, is a pleiotropic alarmin cytokine, most notably expressed at barrier tissues in response to damage. So oftentimes you see this in the skin, in the lungs, and allergy type responses, and alerting early immune responses at those barrier tissues. Earlier studies have shown that it actually plays a role in B cell development, but really there hasn't been a lot done in regards to what this particular cytokine is doing as these B cells develop into germinal center structures. And honestly, for the longest time, there was assumptions that this particular cytokine wasn't expressed in secondary lymphoid organs until this other paper, well, a couple papers that we found, both in my prior postdoc lab and the Ziegler lab, found that blockade of TSLP receptor inhibits antibody responses against allergens, but also that there was a 2016 paper that found that actually if you microdissected out germinal centers from tonsils of humans, especially ones that had hyper IgA nephropathy, they actually expressed higher levels of this cytokine TSLP, and the TSLP receptor is also expressed. And so upon joining Steve Ziegler's lab in 2018, I was interested in kind of digging deeper into what the receptor is doing in the germinal center. So that was our question. What is TSLP doing in the germinal center? And so first thing we did was try to confirm that the follicular dendritic cells in the germinal center were producing TSLP in mouse germinal centers. And so we started this process by just doing some histology and found that in the wild type control, we had overlaying signal between FDCM1, which is a follicular dendritic cell marker, and the cytokine TSLP. And in this particular assay, we included a TSLP knockout control to try to show, negative staining to find any background, but we couldn't find any germinal centers in the TSLP knockouts. So we realized that there might actually be a phenotype here that's broader than what we were initially thinking, and that when we looked into the flow cytometry, we also found that the TSLP knockouts had very few germinal center B cells, suggesting the cytokine was required for the development of germinal centers. So now we had a question we could dig into. Is TSLP required for antigen-specific germinal centers? And then we asked if we could immunize them. If we immunize these mice, can we induce germinal center responses? Because our initial experiments are done with steady-state mice. So upon immunization, we just did a single dose NP-OVA, which allows us to track B cells with this NP B cell tetramer. And we have OVA in case we ever want to go back and do some OT2 T cell work, but we never actually got that far. And we actually used, at the time, what was a pretty new technology, but is the spectral flow cytometry to kind of characterize a large variety of different B cell and T cell subsets. And I just show this now because we now have this technology now here at the flow core, and I think it's a really exciting technology to use because you can look at 50 plus parameters, but usually more than that in terms of numbers of cell populations based off of how you set up your panel. So you can do a very wide scan of a lot of different cell populations simultaneously to identify new populations to dig into a little bit deeper. And so that's what we did here. We started with a very wide 28 color panel that could help us to identify somewhere between 40 different cell types and started digging into a little bit deeper what we're interested in. And so we also tied this particular immunization model in with antigen-specific B-cell tetramers, where we could track over different time points of immunization. So we looked at day 10, 12, 14, and 30 after immunization. We also have a set of ELISpot assays that allow us to characterize high affinity and low affinity B-cell interactions against that same NP antigen. So we can not only look at the frequency of antigen-specific B cells, we can look at the affinity of those B cells as well to see if their maturation through the germinal center is actually improving the affinity to the antigen of interest. So tying these two things together, we just did the first basic experiment of immunizing these mice and looking for antigen-specific germinal center B cells. And we looked at the TSLP receptor knockouts. And this is important now is because we're going to start targeting this receptor on different cell types in the germinal center. So we started with just the total receptor knockout in the entire mouse. Same phenotype, we saw a reduction in germinal center responses, pretty much no germinal centers formed, and a few dots here and there of antigen-specific B cells in those germinal centers. And then when we looked at the antigen affinity responses, total NP reactive B cells as well as the high affinity ones were completely blocked in this particular interaction. All right, and so we want to dig a little deeper into asking, okay, we know that the total TSLP receptor knockout blocks these germinal center functions, but now going and digging deeper into which cells might respond to TSLP most, might be receiving the TSLP signal. So we look for the receptor in different cells in the germinal center. And so starting with germinal center B cells, they do upregulate the receptor, albeit slightly, as they enter into germinal center differentiation. So we use flow markers CD95 and GL7 to mark the naive cells with the negative, double negatives, and at the double positive, those are germinal center B cells. And so we see an upregulation of this receptor as they differentiate into germinal center B cells. And then we look into the T cells in those germinal centers, the ones that are helping the selection process. And we identify T follicular helper cells by CD44 positive, and then they're PD1 and CXCR5 high. And then we had two markers for the regulatory cells that are FOXP3 positive, and the T follicular helper cells are FOXP3 negative. We could use this and actually find that as these B cells differentiated up their differentiation pathway, they upregulated the TSLP receptor. And actually, I'm hiding a little bit of data here that I'll talk about later, is actually the T follicular regulatory cells had a much higher expression of the TSLP receptor. So this is kind of an area of future interest we're digging into. But for the purposes of this particular talk, or this part of the talk, the TFHs had this higher expression of TSLPR. So we decided to kind of now dig in a little bit deeper using one of Steve Ziegler, my mentor's TSLP receptor flux model systems. We could take this system and actually look at independently how is TSLP receptor signaling on T cells impacting germinal center responses. And we do this using a tamoxifen-inducible CD4 crete crossed to our TSLP receptor flux. This allows for us to knock out the TSLP receptor from all CD4 T cells, but do it in a temporal manner. So we're not impacting any CD8 T cells if we do it in the right time frame, or impact any peripheral T cells that are activated before or after the immunization timeline. So we also added in, of course, our TSLPR flux and our CD4 crete controls with that study. We also used two different models that target B cells. One of them was targeting pre-GC B cells, so as they're entering into the germinal center. We targeted using CD21 crete and cross those to TSLPR flux. So this interacts with any mature B cells in secondary lymphoid organs and blocks their ability to signal through TSLP as they're entering the germinal center. But we also targeted germinal centers directly using AID crete. So this is a gene that's expressed in germinal centers that plays a role in the somatic hypermutation process. And so we could actually target B cells after they've differentiated the germinal centers and see how it impacts their ability to mature and leave the germinal center. So starting with the CD4 crete model, we found that same phenotype as we saw with the total TSLP receptor knockout. When we blocked TSLP receptor from CD4 T cells in the germinal centers still failed to form. We saw fewer TFH cells, as well as a reduction in the frequency of antigen-specific germinal center B cells, and a reduction in the number of antigen-specific plasma cells producing high and low affinity antigen-specific antibodies. So using flow cytometry again for the germinal center marker, and then marking out our antigen-specific B cells with the NP tetramer. So this was kind of phenocopying the total TSLP receptor knockout. And so we were thinking, okay, this is consistent. We're assuming that the B cells will look the same. But what was actually interesting was when we targeted mature B cells using the CD21 CRE, we saw the opposite phenotype. So hitting knocking out TSLP receptor on these B cells before they enter the germinal center actually resulted in an expansion of germinal center B cells with a higher frequency of antigen-specific B cells kind of trapped in the germinal center in this particular system. So when we target the T cells, germinal centers disappear, but when we target the B cells, the germinal centers seem to expand. And so we saw increased germinal center size after immunization, as well as the increase in the number of MP-specific B cells. And so what was interesting about this is when we looked at the output of these germinal centers, looking at memory B cells. So I mentioned earlier, we have markers that can identify memory B cells that have transited through the germinal center. And this is kind of pioneered by Mark Schlomchek at University of Pittsburgh. We can identify, based off these markers, CD80 and CD273 are indicators of a lot of T cell help, which suggests these B cells have gone through germinal centers and acquired T cell help. It also indicates that these memory B cells often have a higher degree of mutations in their B cell receptor. Both things kind of tie back to this germinal center transit. But when we look into those memory B cell subsets, and we look into the ones that are from CD21 CRE with the TSLP flock, so again, TSLP removed from mature B cells, they actually make a lot more memory cells, but they don't make as many plasma cells. So it's kind of taking that same pathway where so the highest affinity B cells become plasma cells, the moderate affinity become memory cells, and the low affinity recycle. It seems to shunt out B cells towards the memory compartment and away from the plasma cell compartment. So we're kind of interested in figuring out if this is consistent if you take away the TSLP receptor after they differentiate the germinal centers. And using the AID CRE system, we saw the same phenotype, an accumulation of memory B cells that are derived from germinal centers. And so we had this hypothesis in our head based off of the data we were seeing and some of the primary literature that TSLP signaling we knew from previous literature in dendritic cells activates IR4 expression in dendritic cells. And so we hypothesized that the same pathway through STAT5 will be inducing IR4 expression inside of our B cells. And so we also know in the germinal center itself, in order for B cells to escape, they require NF-kappa B signaling to induce even more IR4 signaling, and that IR4 signaling initiates the signaling cascade that allows for B cells to shut down the transcriptional regulator BCL6 and upregulate the transcriptional regulator BLIMP1, and such that these B cells can escape the germinal center's plasma cells. And so that was the hypothesis. We were thinking that maybe this TSLP was targeting IR4 and limiting the amount of available IR4 to these B cells. So they needed to do more work to be able to get out of the germinal center. And so what we were seeing in the data by flow was that actually TSLP receptor knockout B cells did express lower levels of IR4 at the steady state. And then we could actually do some kind of rescue experiment by taking B cells out of TSLP knockout mice and adding TSLP back in, and we could see this upregulation of IR4 after we added TSLP back to those B cells, suggesting there's some direct signaling there that induces IR4 signaling. And so taken together, we have this interesting cell-specific phenotype where TSLP on CD4 T cells appears to be required for any of these antigen-specific germinal centers to form. But if you look at TSLP on mature B cells as well as germinal center B cells, you need that same signal to let these B cells out of the germinal center. Otherwise, they get trapped in this cycle of trying to get enough signal to become memory cells or plasma cells. And we have some data, both actually B cells and T cells, to suggest that this is mediated by IR4 signaling. And so that was kind of the tail end of that particular story we worked on. But as we continue on and new projects I'm trying to develop in my own lab, as I mentioned before, we actually dug a little bit more into those T follicular regulatory cells, the cells that seem to be shutting down the entire process, but also controlling the stringency of B cell selection. And we know that if we immunize these mice, we use a FOXP3 system, and we can find that actually the germinal centers increase in size again, much like the B cell phenotype. If you take away the TSLP receptor from Tregs in the germinal centers expand. You see a higher frequency of antigen-specific B cells, and they actually class switch into plasma cells more often. So this is consistent with what's known about if you take away these Tregs from the germinal centers generally get larger and make more B cells. Although the mutations we haven't actually looked into in a lot of detail, they may or may not be higher affinity. We haven't looked there yet. So that's some interesting questions we're digging into going forward. So that's one of the major questions we're asking in one of our future projects is how does TSLPR signaling in TFRs, T follicular regulatory cells, impact activity in the germinal centers? We're also looking into the same signal on these memory cells. So I mentioned that these germinal centers, when B cells lack the TSLP receptor, they primarily make these memory cells, but we don't know how good these memory cells are and if they require TSLP to be recalled. And so we found that actually looking at different memory populations, so germinal center B cells versus total memory cells versus memory cells that come out of the germinal center, we see that actually the memory cells that are germinal center derived seem to have the highest level of TSLP receptor. So they kind of upregulate as they continue this differentiation process, suggesting there might be some role for TSLP in those either retention and maintaining those memory cells, or for them to be recalled in the future. We also know, looking into the lungs in the steady state, is actually as these germinal center derived memory cells seed the lungs and develop tissue resident memory responses, they further upregulate the receptor as compared to the ones in the spleen. So we have some literature that we're kind of digging into that suggests that in case of influenza infection, you see in the case of a steady state lung where you have the alveoli up here, with the epithelial layer, there's this region of fibroblasts that surrounds the vasculature called the adventitial cuff. And during inflammation, we actually see this adventitial cuff produces a lot more TSLP, as well as they form these immune structures around the vasculature. And so we have some data, and others have shown that there's normally a TSLP gradient where the highest amount of TSLP is at this epithelial layer, and it kind of decreases as you get further away from the epithelium. But during inflammation, that gradient shifts such that there's more TSLP being produced right around the vasculature. And this is right where all these immune cells are kind of communicating together and undergoing a lot of developmental processes. We did look at some lungs that are infected with influenza and tracked for B cells that express the TSLP receptor. We do see an increase in the number of these cells infiltrating these areas that have high lesions. And so we're kind of digging into another research question, another project asking about how does this TSLPR signaling in these memory B cells impact the establishment, maintenance, and recall of the memory. So do these memory B cells in the lungs usually going to be located closer to the epithelium in the steady state and closer to this adventitial cuff during inflammation? Do they need that TSLP signal locally to either maintain them in those sites or to recall them upon reinfection? Another question that we're kind of digging into is looking at this brand new system called a tonsil organoid, taking tonsils from human patients and kind of making them into single cell suspensions. There is Mark Davis's group, Lisa Wager is the first author in this paper, has her own lab now, developed a system where you can actually generate germinal centers in a dish from human tonsils. So they self-organize upon this culture system, and you can actually add antigens to the system and actually track B cell responses in germinal centers in vitro. And so we actually added our TSLP to the system and found that when we added TSLP to our tonsil organoids, we could actually rapidly generate plasma blasts and plasma cells. So they're actually kind of shunting out B cells faster, kind of opposite phenotype to the TSLPR knockouts. We add TSLP back in, we can make more plasma cells. And so we're going to try to dig around with this a little bit in other projects as well, trying to find out if we can manipulate these tonsil organoids to make plasma cells that have higher mutation frequencies based off of when we add the TSLP. So, and then finally, we have tied to that project some work in collaboration with the Sound Life Project in Seattle, Washington, with the Allen Institute. This group actually collected patient samples from 100 patients, 50 males, 50 females, 50 patients that are 25 to 35, 50 patients that are 55 to 65, and collected single cell time points from 2 years of vaccination from 10 different time points. And they also collected RNA, 5 different spectral panels, proteomics, the whole works. So we were able to kind of dig into this data and had some patients that we were able to identify that had genotypes that had polymorphisms in the TSLP receptor gene, and could dig into the single cell data set and find that these patients actually generated a higher frequency of plasma cells than this is a gain of function mutation of the TSLPR gene. So we can dig into this a little bit deeper in correlation with our work with the tonsil organoids to kind of characterize not only how does adding TSLP impact the germinal center function, but in patients that have excessive TSLP signaling, how does that impact their response to vaccination, to influenza? And finally, another one we have is tied to cat allergic allergies. There is a recent clinical trial targeting TSLP called the catnip trial, in which they provided subcutaneous immunotherapy and added in tezepelumab, which is the anti-TSLP blocking antibody, and found there is a durable blockade in allergen-specific IgE responses. And so in collaboration with Oliver Harrison at Benaroya, we're able to generate a tetramer, B-cell tetramer for FeLD1, the cat allergen, to help identify these B cells. So we're going to dig a little bit deeper into this as well to kind of find out if those same TSLP blocking pathways through biologic therapy also impact specific memory of plasma cells that are cat allergen specific in these patient cohorts. So all that is tied to the TSLP future directions and projects we're kind of digging into. Part of the reason I wanted to tie this in now is since we're getting started and getting excited people to kind of join the lab and maybe be excited about taking on some of these projects. So that's part one, talking about vaccination strategies and trying to improve germinal centers. But I also wanted to talk about what has always been kind of have a near and dear part of my heart, which is the autoimmune germinal center responses, because we're not only looking at amplifying pathways, we're actually trying to kind of figure out what went wrong in a particular patient populations to cause autoimmune disease. Sorry. And so this project actually ties in environmental factors, and I'll get to that in a little bit. But and as this way of looking at autoimmunity as not just a sterile disease that develops, but also all these other factors of our environment, how they impact antibody production. And so the idea, the origin of this particular project started with disease, systemic lupus erythematosus, polygenic autoimmune disease that is characterized by nuclear targeted antibodies. And when we think about the progression of this disease, you have a patient population that has some kind of genetic susceptibility, either through heredity or through heritable genetic susceptibility. And these patients can develop autoantibodies. And actually, one of the interesting things about autoantibodies is a lot of healthy patients that have no overt autoimmune disease are walking around with autoantibodies all the time. But there needs to be an additional hit to actually drive these patients to progressing to autoimmune disease. So autoantibody positive patients that don't have overt disease are in this remittive state. But there's another hit of a loss of immune regulation that can cause active disease states. And in lupus, they call these flares, characterized by fever, particular organ systems being inflamed and targeted, and they can be require some severe clinical intervention to kind of keep the disease under control. And so in the case of lupus, there's a very wide variety of different types of flares that can occur in multiple different organ systems, which is what makes it very difficult to diagnose this disease, is that these patients are very heterogeneous in their clinical presentation. What's interesting is oftentimes there's certain environmental factors that can trigger a flare. And so our goal in this particular project was to identify, and if these flares happen often enough, patients can progress to end-stage organ disease, which require either transplants or very severe clinical interventions. So our goal in this particular project was to try to figure out what drove these flares to occur and see if we could find ways to clinically or to intervene and prevent, stall, or even inhibit the severity of these flares, hoping to give these patients a better quality of life, but also prevent them progressing to this end-stage organ disease as quickly. And so we were focused on a particular gene that is known to play a very crucial role in regulating the immune response, FOXP3. And we know from human patients that cannot express this gene at all, FOXP3 deficient humans develop this condition known as IPEX syndrome, a very severe autoimmune disease that can actually develop in the first couple of months of life and can kill these kids in the first few years without a bone marrow transplant or other very severe clinical intervention. Similarly, in mice, there's a scurfy mouse model, which also lacks functional FOXP3, and mice that have these die within the first 3 weeks of life. And so we're not interested in using these models because you can't really study much with this, and it's kind of depressing to work with mice that are going to die in the first couple of weeks. But we decided to look into this other model that's focused on a different isoforms of FOXP3. So often people just assume that FOXP3 is only expressed in the full-length isoform, but there's also another notable isoform that lacks a second coding exon called the FOXP3 delta exon 2. And we actually have patient data that shows patients that have this mutations in the second exon of FOXP3 that limits them only to making only the delta exon 2 isoform also develop IPEX-like disease. But it's a little bit delayed, it's not as severe, suggests there's some partial functionality of this isoform of FOXP3 that can help retain or prevent immune inflammation. But notably, what's lost in this exon 2 excision are some proline-rich repressor region sites that can bind to other co-regulators of FOXP3, as well as this nuclear export signal. These things are things of active interest we're kind of digging into now, looking at mechanisms, but you do lose some functionality of the FOXP3 protein, but it's still able to transcribe certain genes. Well, most genes, I should say. So to develop this mouse model, Barajou, a former postdoc in Steve Ziegler's lab, actually generated these mice where we excised out the second coding exon of the mouse FOXP3 gene and confirmed by sequencing. And actually, we have reagents that allow us to identify the different types of FOXP3 because we have one antibody that binds across the exon 2 junction. So this can bind to both isoforms, the full length and the second, the delta exon 2 isoform. But we also have an isoform of, antibody that binds the N-terminal region of the FOXP3 gene. So we can identify just the delta exon 2 isoform of FOXP3 as well. What's also exciting about this particular model is that FOXP3 is in the X chromosome. And so we can use that, and we'll talk about this in a few minutes, to manipulate these mice to generate females that are hemizygous for FOXP3. And because of X chromosome inactivation, we can express both types of FOXP3 in the same mouse, but they're only going to, the Tregs they make will only be wild type or only delta exon 2. So we can do direct in-mouse comparisons of the same Treg populations. But what I wanted to note was actually when we looked at these mice, we expected to see a similar IPEX phenotype, but they did not develop this. These mice lived perfectly happy mousy lives for 2 years, and they only had some inflammation in their lymph nodes and slight inflammation in the spleen, but they didn't develop this overt severe autoimmunity that we're seeing in humans. And so kind of left us with the question of what's happening. So I came into the project looking into germinal centers, and I was interested in those regulatory T cells inside the germinal centers. And so again, mentioning that upon immunization, you see these germinal centers expand and contract. And at that contraction phase, you see an influx of FOXP3 positive TFRs. And those are the ones that seem to be shutting down the germinal center response. And when we looked at our FOXP3 delta exon 2 mice, we actually saw that those germinal centers were chronically enlarged. And we see a greater influx of these Tregs. So they're entering the germinal center, but they're not able to shut it down. And further yet, we could see that, so same thing. We could see that the number of germinal centers per area wasn't really controlling the structure. So we wanted to ask if this increased the frequency of autoantibody titers or any antibody titers in general. So they have enlarged germinal centers. They're making more of these B cells. This should, in theory, result in more antibodies. And what was interesting was we saw an increase in the amount of IgG. We saw a 20,000 times increase in the amount of IgE, which is very strange because usually IgE is one of the best regulated antibody types in the periphery, and oftentimes it's barely detectable in the serum. But we're detecting this at basically levels that are similar to some of the IgG subsets. So we're pretty excited about this phenotype. And we also were able to see increased numbers of IgE producing cells in the spleen in every lymph node we sampled. So we wanted to ask, first that we have a ton of this IgE being produced in isolation. We're not immunizing these mice. We're not, they're just generating a ton of IgE. So what is it targeted to? So in collaboration with PJ Utz and Sarah Chang at Stanford University, we ran an autoantigen array with our IgE and kind of ordered these based off of the p-values of the hits and found that actually keratin and skin antigens were the highest targeted targets by IgE. And most notably, the highest hits are all antigens that are released by keratinocytes after UV damage. And so this kind of pointed us in the direction of asking about these environmental factors again. If we can give UV light damage to these keratinocytes and they release these antigens that seem to be targeted by our IgE, can we induce a lupus-like flare, which is similar to what you see in the disease patients that have lupus. They have exposure to a lot of UV light, get a sunburn, and they have a whole body flare that happens shortly after. So we designed this, coming back to this idea again, where environmental factors are triggering this disease activation from a remittive state to a flare state, is IgE, and IgE that targets keratin mediating this process. So we decided to take some mice, we shaved 2 sites of their back, and we tried to model this UV damage response by treating one side of the skin with UV light, and then the other side was protected with foil. And so we could do this over 6 different doses and induce this inflammatory response. And this inflammation was exacerbated further in the delta exon 2 mice, not in the wild type mice. So we gave a low enough dose to have a mild enough response in wild type mice. We didn't see much of a difference, but we did start seeing phenotypes of skin damage occurring in the delta exon 2 mice. Further yet, when we looked into this draining lymph nodes downstream of the skin, those germinal centers that are enlarged in the delta exon 2 mice were blew up quite a bit, generated a lot more IgE producing B cells. And we could also see the IgE coming back and targeting the skin. And what's notable about this is actually these are the skin samples and the lymph node samples are paired from the same mouse. So the untreated side had lower IgE, the treated side had a lot of IgE being produced. And when you look into the skin, the IgE was only depositing in the damaged skin site and not in the undamaged skin site. So this is pretty exciting, giving us the idea that there's this localized effect that's generating skin reactive IgE. Now, the next question was, what is the IgE doing? If it's going to the skin and just binding there, it should be activating these type 2 responses that didn't really match up with what we're seeing in some of the data. And so we dug into the literature a little bit further and found this interesting phenotype that's been kind of published called betosis. So much like neutrophils that net and throw out their DNA contents, basophils do the same thing here. So here's an electron micrograph of a basophil that's throwing out its bets into the tissue. And you can actually use microscopy to identify these bets. And one of the markers we used was MCP8 to identify them in the skin. And so we were asking I should take a step back. The basophils undergo betosis when IgE is on their surface and cross-links. It is different from when basophils degranulate because we don't see the same markers of degranulation. So this is an independent process that's occurring seemingly in response to certain inflammatory cytokines that have yet to be defined. We're still working on that. They can generate these bets in vitro, and the bets do not occur if you swap out the IgE for another antibody subtype. So when we looked into the skin of our UV treated mice and our wild type controls, we saw a few of these green dots, which are the MCP8. This was a marker of betosis. And if we zoomed in, you can see that their DNA is kind of splayed out a little bit. We see an increased number of bets in the wild type mice when they're treated with UV light as compared to the untreated side. But you can see there's a whole lot more of these guys generated in the delta exon 2 mice. So they're covered in these bets. And actually, when we kind of looked at the 3D structure of those, we could see that they span quite a far distance to drive this inflammation at this site. And so quantitating this by microscopy, we could see there were about 3 times as many bets that were forming in the UV treated skin of delta exon 2 mice versus the wild type controls. And so we wanted to ask if this is driving some local inflammation down the road, downstream. And so we just took whole skin and did RNA sequencing, identified certain gene signature pathways. Interferons, of course, came up. There's tons of DNA floating around in their skin. And we also did some confirmation Legendplex kits to identify any kinds of actual levels of interferons in the skin. We did see increases of type 1 interferon in both wild types and delta exon 2s, but actually what was really different between the wild type and delta exon 2s was the interferon gamma. So this is a bit of a strange phenomenon. We're seeing tons of IgE, but we're also seeing a lot of interferon gamma being produced at the same time. And in the type 1, type 2 categorization we typically have, those things usually should inhibit each other. But we're seeing both of these in the same context in this particular phenotype. So this is one of the areas we're kind of digging into a little bit deeper, is trying to figure out how this interferon gamma signature is being produced in the presence of a ton of IgE. And so kind of bringing our model together, in a wild type mouse, we have this model that suggests that UV damage drives this germinal center response, and you see this modest amount of self-reactive IgE that activates basophils to respond at the tissue damage site. In the case of our autoimmune prone FOXP3 delta exon 2 mice, we see a whole lot more germinal center responses. Actually, this should be enlarged, but more IgE that coats more basophils. And now all those basophils go to the skin and undergo betosis and release their nuclear contents and drive further inflammation locally at the site of UV damage. So taking this forward into what I'm trying to dig into further in my lab, one of those is digging into how Tregs in the skin impact the epithelial turnover and maintenance after UV damage. Another is how does that skin reactive IgE mediate inflammation? We're still working on that for this particular paper. But another one I'm going to talk about in more detail in the next few slides are looking further into how do the T follicular regulatory cells inside the germinal center drive this process. We're specifically looking at this ton of this extra IgE being produced. Why is this defect in Tregs making IgE specifically in such a high amount? And so actually some work from Kristen Weinstein, one of the graduate students in Steve Ziegler's lab. As I mentioned, she was using this clever strategy with X chromosome inactivation in females. We could express both types of Tregs in the same mouse and then sort them out and look at them in a paired RNA sequencing statistics algorithm. So we're able to do this with TFRs in the germinal center in the steady state and pulled out several different pathways. We're still digging through the data. Related to cell cycle was mostly increased in the TFRs that express delta exon 2, but we see a lot more downregulated pathways that are associated with type 2 immunity, which is very strange. Again, seeing reductions in IL-4 and IL-13, which we would expect to see more of this because there's so much IgE. So this is kind of a perplexing phenotype we're seeing in the steady state. But I should also mention we did this in UV treated mice. And once they're UV treated, we actually see kind of a flip in a lot of these pathways. So this might be just a maintenance versus active inflammation kind of phenotype difference. We're digging into this further. This is still a point of continued work. But one of the other interesting details is that in those scurfy mice, the ones that lack any FOXP3 at all, you also see this super high level of IgE in circulation. So we wanted to try to see if it's just total Tregs knocked out, or if delta exon 2 expression in all Tregs is driving this IgE phenotype, or is it just specific to the TFR compartment? So we tried to find ways to actually specifically isolate delta exon 2 expression to just that compartment. And so one of the ways we can do this is by using this mouse model that express FOXP3 Cre and BCL6 FLOX. So BCL6 being a transcriptional regulator of germinal centers, it's also expressed in TFHs and TFRs to maintain that differentiation state. So if you cross into a FOXP3 Cre, any Tregs that express BCL6 FLOX or BCL6, meaning TFRs, can't be generated. They lose their BCL6, they cannot generate the TFRs. So this is effectively a TFR knockout mouse. And we see this showing, when we look by flow cytometry, they basically generate almost no T follicular regulatory cells. But we can take this model and make a mixed bone marrow chimera and with the TFR knockouts and replace the TFRs with whatever it gets mixed with. So if we mix the bone marrow from TFR knockouts with wild type, the TFRs will be wild type. If we mix the bone marrow with delta exon 2, the TFRs will all be delta exon 2. So we can selectively express delta exon 2 in the TFR compartment. So using this model, we actually did this. We generated TFRs that are only delta exon 2 versus wild type controls, this bone marrow chimera system, and we saw the same phenotype. So if delta exon 2 is limited to the TFR compartment, we see enlarged germinal centers and more of this IgE in circulation. Got really excited until we started looking, going to the spectral data. You can do a lot of stuff. We also looked into the TFR compartment, and we couldn't find our TFRs from the delta exon 2s. They didn't survive as much. So we have a couple of things we need to do digging into this a little further. So so looking into the TFR compartment using our dual labeling system, we can see that the dual label is present in the wild types, but you should see just a single label in the delta exon 2s, but we're not seeing those TFRs reconstitute the TFR compartment. And so we just looked at the TFR knockouts. Do they have increased germinal centers and increased IgE? And they do. So looking in just the steady state of the TFR knockout mice, they have enlarged germinal centers and more IgE. So this is one we're kind of digging into a little further, is what happened to those TFRs. So we have two hypotheses. One is the delta exon 2 Tregs were outcompeted by the wild type Tregs before they differentiated into TFR cells, which is actually supported in our previous work, where we found that after thymic differentiation, there's a skewing that 10% of the Tregs in the periphery are delta exon 2, and 90% of the Tregs in the periphery are the full length. Somehow they're getting more survival signals, and we suspect that's through CD25 or IL-2 signaling, which is known to keep Treg sustained. So that's one hypothesis of what's happening. Another is that once these Tregs enter the germinal center, they rapidly de-differentiate and become T follicular or become X Tregs. And so this is also supported by some of our previous work, where we use this dual labeling system to identify T cells that had once expressed FOXP3 and then had lost it. And we found, they call those X Tregs. And we found more of these X Tregs in the delta exon 2 expressing mice. But we haven't asked this question inside the TFR compartment, inside the germinal center. Do we see the same occurrence where T cells become Tregs, enter the germinal center, lose their FOXP3, and now become effector cells that drive more autoreactivity? So this is another hypothesis we're kind of digging into a little bit further in some of the future work in our lab. And so with that, there's a ton of people to thank, most notably all the people from the Ziegler lab that I worked with when I was in Benaroya Institute. Most of these people on here are from Benaroya, but I obviously hope to generate new projects here at UBC to kind of fill this out with UBC folks. But most notably the genomics core, and I should give a lot of praise to Christian for generating some of this RNA sequencing data, developing this mouse line to help us get going. Some other notable thanks to Aksa and Florence, who did a lot of work with this throughout the years, getting this project off the ground and running, and all these collaborators for doing different components and helping out with either the TSLP work or the UVB project. And with that, I'd be happy to take any questions.
Thanks very much for that, Philip. After the questioning, Caitlin will be really angry if I don't make 3 announcements. So are there questions? Go ahead, please.
Thank you for the great talk. I have a really naive questions. For the TSLP, you say in the mice there's a defect in the germinal center like development. I just wondering, does the TSLP also affect the CD4 T cell, their selections, for example, from the thymic, and not only for the follicular helper Treg cells, but also in the circulating, like general in the Treg, does it also affect it as well?
Yeah, so I don't, in this particular project, we didn't specifically look at like thymic selection processes, but the lab had previously done some work with that and didn't see anything that was notable. But I should mention that a lot of the technology we used at the time was pretty not new technology, so it might be worthwhile to kind of dig into some of those defects, especially with the whole knockouts. The reason that was also part of the reason why we used the inducible system was we were concerned there may be some defects in some T cell development, and we did know there were, earlier on that may impact the phenotype. So we used the tamoxifen inducible system so we could just label to knock it out right before immunization, and we could only do experiments with this mice for 2 weeks, because that was around the time when you started seeing any thymic emigrants repopulating in the periphery that may have also been hit by the Crete. So we did actually consider that might be a phenotype, but we don't have, in this project, we hadn't specifically looked at that. So that's worth digging into further.
Yeah, always something new to see there. So really strong BCR signaling and TSLP receptor signaling seem to have the same effect on the fate of germinal center B cells, driving them more towards plasma cells than memory B cells. I was wondering about the interplay. Does the strength of BCR signaling regulate the upregulation of the TSLP receptor, and then how much TSLP signaling the B cell gets?
Yeah, so we did some work with the tetramers to find that actually the antigen-specific B cells did have slightly higher TSLP receptor expression of their cell surface. I'm not sure if that's driven by that initial naive B cell activation or if it's within the germinal center itself. We have some better tools with our spectral panels to interrogate that a little bit further. Which, yeah, I think that comes down to the molecular signaling pathways of how does BCR tie to the promoter of the IRF4. Yeah, I think those are, yeah, good questions that we haven't really answered, I think, well enough for my liking, but yeah.
I can use that like hell-specific B cells and the whole panoply of different bird lysozymes.
Yeah, so you reminded me, that's the second part I was going to mention, was we have in some of the grants using B18 knock-in. So these are NP-specific B cells. Sorry, B cells that express a B cell receptor that is already reactive or specific to the antigen we used. So we kind of give them a cheat so they can get in faster and they can outcompete the other B cells in the germinal center. And that can probably help us to interrogate that question a little bit further as well.
Very interesting talk. Is there any role for interleukin-7 in germinal centers, and does it compensate even partially for the TSLPR knockout?
Sorry, I missed the first part of that question.
Interleukin-7, is there a role for it in germinal centers?
Yeah, so the germinal, we did actually get grant or comments asking us to check IL-7. IL-7 receptor is upregulated in those germinal centers. Actually, it's higher on plasma cells, I think was what we saw. And we did see some on memory cells. So there's something going on with IL-7 signaling locally in that structure. We haven't done it justice digging into it further, but there are other data out there, especially in B cell development, that really hammer down that IL-7 plays probably a dominant role in the development of B cells early on in the bone marrow. So it's probably playing something, doing something up there. Yep.
Nice talk, Phil. I was wondering for the FOXP3 delta E2 mice, if instead of doing a UV damage to the skin, if you vaccinate those mice with an antigen, for example, on the skin, and with some like Th2 polarizing adjuvant or cytokine, will you be able to observe any different form of IgE that's specific to that antigen?
Yeah, to use a Steve-ism, when we give them allergies, they melt. Basically, they get a really bad allergic response if you give them allergy challenge in the Th2 priming system. That's actually part of the reason we bifurcated projects is some other groups that we were working with are kind of pursuing that in some more deep depth in the actual allergy system, kind of in some more depth. However, in the germinal center side of things, we've also immunized just by IP and using type 2 that, yeah, we do see a very strong germinal center response, a very high NP reactivity, but we have to dig further into the BCR maturation through somatic mutation because like the TSLP data, we had some ELISpot data that didn't really show they're actually affinity maturing. They're just, you have more low affinity NP reactive B cells, but not a lot of high affinity ones. I think that's a better question to ask with this TFR replacement system, if we can get it up and running, because that'll be a more selective way of just targeting what's in the germinal center. But yeah, I think your questions are right on for stuff we're definitely interested in doing. And yeah, thanks. Cool. Yeah, thanks. Great question. Yeah. Yeah.
Oh, hi. Really elegant work. Nicely done. I'm just out of curiosity, does TSLP receptor signaling do anything interesting on early life B1 B cells that are germinal center independent? And I'm very curious about that. And, you know.
Yes. So I did this in the deltas actually, haven't yet done it in some of the TSLP project where I have, going back to spectral, we did a 30 color panel looking at all the different B cell subsets in the peritoneal cavity, as well as the bone marrow, spleen, thymus, lymph node, lung and try to just characterize any defects in B cell development. I think that would definitely be worth doing, especially peritoneal B cell populations with the TSLP knockout systems. We actually got rid of the CD21 crease, which probably would be the best system to use for that. But maybe just even looking at the TSLPR knockouts in general and just seeing if there's any defects in B1 B cells to get started, and maybe justify going back to the CD21 crease in the future.
Yeah, and I would just make a pitch there for you to take a look at mice where you've kind of skewed the biome towards a pro-immune phenotype, pro-allergic phenotype, because we find that if we put mice on antibiotics, they really are prone to allergies, and I'd be very curious to see how it influences those guys.
Yeah, and so there's we actually, the NSERC grant we put in for the TSLP project related to TFRs, we're really interested in at how the defect or loss of TSLP on the TFR compartment, how does that impact gut microbiota? So I think that would tie in pretty well with our questions of, we know that that TSLP signatures at the barriers in the gut are really important for maintaining that microbiota. So it wouldn't surprise me if there are some defects or some changes in microbiota specific B cell responses as well. So yeah, the T cells are just the lower hanging fruit to get started with, but I think, yeah, we can definitely talk more offline about the prospects of doing more with B cells and microbiota. I think that'd be a cool question too. Okay.
Good to go, Jamie.
Oh, thanks. Yeah, that was a beautiful talk, and you have definitely done a lot of digging. Anyway, yeah, I just wanted to ask this kind of an off the cuff question here, and that is, have you seen any involvement, especially under chronic conditions, in your UV light model, for instance, of eosinophils and those kinds of infiltrates?
So we tried looking, so the original goal was looking for nets. And I thought that was going to be the nice, clean tie up for this paper, and that would be done. Nets didn't show up. Eosinophils are there. We haven't seen any changes in activation markers different from the wild type controls or any indication. I mean, they are coded with IgE, but for some reason, they're not getting readily activated. It's the basophils that are, we've looked at, mast cells as well. Those are the ones that everyone's been kind of most perplexed by. Mast cells are there. They're just not any different than the wild type controls and all the different metrics we studied. So eosinophils in that same category. I think it depends on what kind of challenge system we use. I wonder if we added in some type of a foreign antigen with UV to kind of simulate the MC903 plus antigen, we might get something that looks more eosinophil driven. But for the life of me, I don't know what's going on with eosinophils and why they don't show phenotypes, whereas it's just basophils. This is one of the things that's kind of holding up the paper, is that last mechanism detail we're trying to tie into, and we're poking around and trying to figure that out.
Thank you.
Yeah.
Phil, yeah. I'm unclear about what other ligands a TSLP receptor may have, and simply does tezepelumab, the TSLP antibody, have similar effects as the receptor knockout?
Yeah. So Tezi trial did seem to show. That's why we were kind of digging into the germinal centers. We can look at in the PBMC circulating T follicular helper cells as a proxy for germinal center activity. We could also put Tezi into our tonsil organoids, which is what we're planning to do to answer your question of, does that show the same TSLP receptor knockout phenotype? Other ligands for the TSLP is kind of a funny question that I think is in the back of a lot of people's minds in the field, because there are some phenotypes that's slightly different between TSLP knockout and TSLP receptor knockout. And part of the reason why I did it in both systems was because I wanted to confirm that this is like cytokine specific to what I'm looking for. But there might be some other ligand or other receptors that TSLP binds to. I'm not aware of any other ligands that bind to the TSLP receptor. It does form heterodimers with the IL-7 receptor, which is part of the reason we were at. So that the IL-7 signature through SAT5 is very similar between TSLP and IL-7 alone. But in regards to other ligands that can bind to that TSLP receptor. I'm not aware of any of those, but the other flip side of that question of what else can TSLP bind to is something we still, I think there's an open question to that too. But yeah, so we dug into using the receptor flox mice in part because that's a more defined question we can dig into with clean air data. But yeah, that might also be a good question to ask in the future.
Thank you very much.
Yep.
Are there more questions? And with that, if you will indulge, I have 3 announcements, and you're invited. The Center for Blood Research is having a skating hour over at the arena on March 7th. Please sign up by March 4th. Mike Gold. Norman Bethune Symposium, Center for Blood Research, April 10th. And the Center for Blood Research Spring Magazine is requesting cover photos. So see Caitlin about that. That's all I have. And sir, thank you so much for joining us.
Yeah, thank you all for coming. I appreciate it.